chemistry. We just we can be talking about it in terms of what the chemical makeup of the genes are, how are enzymes structured, what are the enzyme concentration, how can you manipulate those? In pathology, basically most of the pathology, especially if it's viruses, are driven by taking over the host genetic architecture and then expressing as form of in form of phenotypic characteristics that are known. So if you have like a viral infection, whatever you're seeing is because the virus is able to replicate within your body and multiply and then show those effects. Even in microbiology, for example, in yeast making, they are already working with take out yeast or make yeast much more potent in what they do, or that is genetic improvement, or they are cloning the yeast. For example, the the human genome human genome that was published about 25 years ago started by using like bacteria accrested chromosomes. That's how they clone uh, these genetic strands, and then before they were able to like uh, sequence them using Sanger sequencing. So, as I was saying, genetics is rarely looked at just one. Genetics is mostly uh, a tool for understanding more of like life sciences. As a result of that, and understanding that most of what I'm talking about, I'll link it, for example, to genetic diseases, because most diseases start as a DNA change in DNA, which alters protein function, and then proteins or enzymes, which are basically the drivers of physiology in the body. So anything that changes the protein has a detrimental effect on the body. Also, we're talking about antibiotic action. How do you use antibiotics? So, for example, antibiotics was discovered as a result of a mistake or by chance. But then, why did antibiotics work so successfully in human beings? It's because it's taking advantage of our difference between the 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 bacteria and the virus. No, no, viruses are rarely affected by antibiotics, but bacteria, which is quite different from humans. So at this point, I'll be talking about prokaryotes and eukaryotes. And then most bacteria are prokaryotes, and humans, or like you studying humans, are eukaryotes. There are fundamental differences between prokaryotes and eukaryotes. I'll be touching on this. And then I'll talk about vector control and some of my studies during my PhD and some of my studies in my postdoc, where we can leverage genetics to be able to do good in the society, not just do good, understand changes across our environment and then adapt to those changes as well as uh, prefer solutions to them. And then lastly, I'll bring most of what I'm talking about to things that matter to you as a clinician going forward. How are you going to have a look at the disease and then, for example, attempt to solve it? Some of you are not going to end up as medical doctors. Some of you might be lecturers, teachers. Some of you might work in pharmaceutical companies. We have medical doctors who are part of our work. So some of you could be like researchers working in labs. If you're doing that, the fundamentals of whatever you're doing, unless you're not working with humans, you need to understand genes. So, for example, in my department, there is the genetic disease. Most of our diseases are genetic disease, but you'll be surprised that we're using small molecules, or called small molecules, things like paracetamol is a small molecule, molecules that have they're able to act as ligands that goes to enzymes and then either focus in that pocket to knock out the enzyme or make it more potent to be able to elicit a physical response. So understanding chemistry is good, but understanding biology is very fundamental. But then understanding that enzymes are very unique and central in making uh, human function as they do is very good. But then beneath that enzyme is the genes that made it. Any changes in genes is going to affect how enzymes are structured. So as we go along, I'll be not forcing, I'll be encouraging you to think beyond uh, 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 I'll be encouraging you to think outside of the box, sorry. So that's how whatever you're looking at, how is this a genetic disease? Does it affect whatever I'm doing or whatever I'm seeing? How do you now in drug discovery where I do my job, how do we leverage these changes to be able to create drugs to either amplify the change we see or reduce the change or sometimes knock it off. So I've been introducing things like CRISPR, I've been introducing things like polymerase chain reaction, I've been introducing things like cloning, some of those things that we use as tools to help us understand genes in its entirety. So if you have questions, can you ask? Or anyone, feel free. This is a point where I'll migrate to the next stage. Anybody have any question? Okay. Okay. So, like I said, we have three lessons. This is a three-part lesson. The first lesson will be focused mostly on DNA to RNA, look at transcription, gene vocabularies, transcription inhibitors, and there I will introduce. So for these three lessons, I'll be introducing some of the work I did as part of my PhD and what I currently do, just to give you a real life context about how these things are. So it's not just in the textbooks, but see what people do with it in real life and how it affects your day-to-day lives. And then the lesson two will be the RNA to protein. How is RNA transcribed into proteins? What are the enzymes involved? What are the different mechanisms for this happening? And then I'll introduce the concept of genetic codes, translation, or even antibiotics, and why antibiotics, for example, are so successful. And then for this, I'll talk about Wi-Fi gene families as part of my PhD work, and how, for example, gene families can be used to differentiate different Wi-Fis and how to respond to them. I even target them in terms of insecticides use. And finally, control and consequence. I believe this is the ones that medical students should be more interested in. So it is all good understanding DNA to RNA to protein. But if there's any change in any of this process, can someone mute, especially the person in tech? So if any change happens from DNA to protein, it affects what you see physiologically. So I'll be talking about operons, regulations, mutations, and genetic disorders. So, so the first lesson from DNA to RNA, this will be very simple. By the end of this lesson, hopefully, if you didn't, maybe it's a refresher to what you already know. By this time, you'll be able to define key terms such as genes, what is an allele, what is a locus, what is a cistron, what is a promoter, open reading frame. These are things I use basically at work randomly without thinking about it. But if you've not been introduced to it in its entirety, it becomes harder to grasp it. So your job in my job is to put all this in context in a good story, which allows you to, when you read about it, you tend to use it more in your vocabulary. So understanding cistron, I don't have to explain it to my colleagues. We all know what it is. If I say I'm looking at as a sequence, it looks like it's polycistronic. I don't have to explain what polycistronic means to the person because he already understands it. So if you're able to understand these key terms, then you can begin to live, as in migrate from understanding what you're looking to actually applying it and using it in your day-to-day lives. The second one is describe transcription. So I understand this must have been introduced in your other classes. But in this, I'll just go through most of the process transcription, but mostly I'll be tying some of this transcription to effects or what happens outside, especially in the drug industry. How do we target this process? And then how do we leverage this process to make drugs? I'll talk about initiation, elongation, and termination of RNA synthesis. And at this point, I'll talk about some of the concepts of transcription that we all know about. I also compare prokaryotes and eukaryotes. I talked about cistronic and polycistronic and monocistronic. What is the difference between prokaryotes and eukaryotes? It's a DNA. Okay, the DNA coming from, for example, bacteria is different from the DNA coming from humans, not DNA in itself, like the genetic makeup. And then the cell of a prokaryote is quite different from the cell of the eukaryote. I'm sure you guys have known about it. In this lecture, I'll be talking about how does those differences affect their different mechanism of creating proteins. Why is it that a bacteria will make protein in a different way than a human? So in doing this, for example, if you're trying to make insulin in a bacteria, which is how insulin is made anyway, you need to understand how a bacteria will take that gene that you inserted into its cytosol to be able to generate the protein you want. So understanding the bacteria is just as important as understanding our own cells. And then here I'll explain inhibitors and why selective inhibition works. The whole concept of antibiotics is, is hinged on selective inhibition. So basically, a drug or an agonist being able to go into the system and then selectively knock out something. So, for example, if you're fighting in a war and there's no way for you to know who your friends are, then no one's going to give you a gun, right? Because you've killed both the enemies and the foe. So if you take an antibiotics and the antibiotics has no way of differentiating between your cell and the cell of the bacteria you're trying to kill. For example, you have like a lump in your eye. How does that drug you take in your mouth know that the bacteria it should kill is the bacteria causing that lump in your eye and not your own eye cells or something? Or in fact, how does even the drug you took in your mouth affect, for example, the sore you have in your legs? So that's basically pharmacokinetics, how drugs travel. But then when a drug gets to its point of action, how does it know to discriminate between affecting the real cells and then the cell of this target organism? So the whole point of drugs, in the industry which I work in, and then antibiotics is hinged on the ability of those drugs to target the differences between prokaryotes and eukaryotes, which is why this is very important. And then finally, I'll be applying it to real data, giving you a story of how I came in across West Africa and then collected samples and then went in to sequence these genes. So we talk about genes, but what genes are? Genes can be boiled down to just ACTG, sequence of ACTGs. If you understand it that way, then genes starts to become very big. So if you're able to like conceptualize it, that genes, it's actually made up of like your DNA, right? Your genome, right? You can make out gene from your genome. But if you know that you can actually represent genes as ACTG and series of these strings, it becomes easier for you to have it in normal conversation, but also articulate whatever question or you're asking an exam or solve a problem in real life. Do you have any question? Okay. Is there any question? Sorry. So
No,
before we
sir.
start, sorry, you said something. Okay, you said no question, right? Okay, that's all right. So I'll introduce you to six vocabularies, but it will be important to pay attention to what I say because I've been working in this field for a long time. I might be using vocabularies that I think I guess you know or I forgot to have been introduced to you. So if you hear anything I say that you don't understand, let me know because there might be a story behind it, and then I can give you the more context I'm able to build across the knowledge base. It's easier for you to go into your learning jar or your cell biology textbook and then read it with understanding. So this is not trying to help you in a way. I'll provide a lecture notes as part of this, as well as this presentation. But the whole point, I will have done my job very well if you open a but molecular biology of the cell. That's the textbook I'll suggest you read. You open it and go to the genetics part and you read it with understanding. Understanding. It might be more in depth, but you read it with understanding, knowing, this is actually very simple. It starts with understanding the vocabulary. So, for example, gene, I'm not going to describe gene, but I'm guessing all of you know what a gene is. A gene should have an open frame. It should have a start and an end. Most times it has a function. Sometimes there are no function. A gene is basically made up of a DNA string that has a reading frame that can be translated from what it is into like a polypeptide or a peptide. That's a gene. But then alleles, uh, alleles starts to make sense when you think about um, mm, about the fact that genes comes in pairs. Can anyone tell me what it is? Like diploids, right? You know what I mean by diploid and haploids, right? So there are some organisms that are haploids. There's some organisms that are diploids. When I mean diploids, so if we have a chromosome, like you're born as a human being, you're made up of the chromosome from your mother and from your father. So there is two copies of every gene you have in your body. There's always two copies. One of that copy is called an allele. Is that clear enough? It is these differences in alleles that variations differ. So if you bring two twins, not identical twins, because identical twins are almost identical to like 98% or 99%. Yeah. But alleles are basically where those variations are. I'll talk about alleles in our third lecture when I introduce the concept of GWAS, which is genome-wise association studies. You go down to alleles. Sometimes you're working with the haploid genome or you're working with the diploid genome. And then at that stage, allele starts to make sense. But just understand it there so that when I introduce it, it becomes more conversant. Then cistron. Cistron is basically that unit of coding for one peptide. So if we have 15 or 20 DNA sequence, so ACTGTGGAATCGT to the end, that sequence of of DNA that you're able to transcribe into an RNA, which can translate into a protein, is called a cistron. Now, if you listen properly, you hear me say words like monocistronic and polycistronic. So in bacteria, for example, uh, bacteria are known to be polycistronic in that, for example, if an enzyme I'm guessing you guys remember the Meyer-Hof's pathway, but like glycolysis pathways, right? Is that familiar to you? I asked the question. Maybe you can nod or say hi. You're familiar with the Meyer-Hof's pathway, the glycolysis pathway, right? So the process of making energy from glucose. That's familiar to you guys in biochemistry, right? Okay. This involves the interplay of several genes. I can't remember them, but some dehydrogenases, some genes that converts the glucose into something that the body can do, ultimately getting to the end, which is like, I think, ethanol. So these are suites of genes. In humans, these genes are rarely in one place. So, for example, the first enzyme, the first gene, the gene that makes the first enzyme might be in, in chromosome 2 of the human genome. And then the gene that makes the next enzyme might be in chromosome 3 of the human genome. This this process means that a lot of genes that is needed for one process is not stored in one place. This process is the same as monocistronic. So it's unlikely that one gene can give rise to several polypeptides. But in bacteria, for example, the same glycolysis process, if a particular process needs like 5 or 6 genes, in bacteria, for example, you notice that one stretch of DNA will encode that 5 or 6 genes in its own. And then the reason for this is that in bacteria, because bacteria are known as simple organisms. When we mean simple, we don't mean like simple in that it's easier, not like they have easier genome because I've assembled bacterial genomes. There are some are quite big, but it's simple in that they have less complexity. They cannot afford complexity because can you tell me the difference between can anyone tell me okay you don't have access to charts the difference between a bacterial the key difference between a bacterial cell and a human cell or diploid cell. The key difference between a bacterial cell and a human cell is that the human cell or other eukaryotes have an enclosure around the nucleus, while bacteria don't have that. As a result of that, they're built to be simple by nature. So if they need 5 genes to drive a process, they put all those 5 genes together. And then when you are transcribing those 5 genes, you can transcribe them at the same time. So it helps with genetic regulation. So to achieve easier and simple genetic regulation, bacterial genes can be polycystronic. That means it has one gene stretch has the ability of making multiple genomes. So someone asked a question. Sorry, let me check. Okay, sorry. So feel free to write anything there, the class rep. So in case you need to tell me about anything, maybe send the message there. I'll say it. And that's for cistron. I'll be talking more about it when I start to describe the difference between the bacterial genome and a human genome. I'm using that as an example. I could say eukaryotic and prokaryotes, but just to give you a context and bring it down to what you have. Bacteria is what we all know. It's like the cause of most like 80% of infections that we know about. Promoter. So within the gene, not everything is transcribed into a protein. So there's a promoter region, there is the repressor region, there is the introns, there is the exons, there is the end, there is the so a gene has a lot of different. So even if you have like 20 or 30 DNA sequences, it's unlikely that everything gets into a protein. Some of them act as promoter. So in essence, promoters are basically the DNA region where this RNA polymerase, which facilitates the creation of RNA, recognizes and then attaches to in order to do the DNA assembly. So, for example, if you want to make sure that your DNA does not work, it's not made into a protein. For example, if I was is part of my work, I want to I have a gene that is causing a problem and I want to silence that gene. I simply write an antisense to that promoter sequence. So when I said something about azos, I was working on azos. I'm working on antisense oligonucleotides. I write an antisense that recognizes that promoter. And when it goes bind to that promoter, it prevents the binding of the RNA polymerase completely from attaching. And once RNA polymerase does not attach on the promoter, then that gene doesn't get transcribed. And if that gene doesn't get transcribed, doesn't get translated into a protein. And if it doesn't get translated into a protein, then that enzyme doesn't exist. As simple as that. So that's how I'm trying to connect what you're understanding now to what we do in real life. So it is basically biology. If you understand biology in its basic sense, then you're able to like leverage on it and use that to advance science and medicine. Then open reading frame is simply a chunk of DNA that has a beginning and an end. So when I mean the DNA, like AUG transcribes for methionine, and then TAA, TCA, and then something else, I've forgotten the name. Three sequences transcribes as end or stop codon. I will introduce the concept of codons in this lecture about, yeah. So looking at that DNA is not just a random ACT, GTCG. They all make sense if you consider them as triplets. So ACT, ACT, CTA, TCC, TAA, and those are called codons. And it's those codons that the tRNAs recognizes in order to attach a given protein. Now I'll introduce the concept of genetic code, maybe in this lecture or the next. So you see how all this is intertwined. Now, I just by talking about ORF, I'm already talking about codon. So I was describing ORF, but I needed codon to explain it. So an ORF and a codon. So codon is just the three base word that is recognized by the tRNA that represents a given amino acid. Now, how many of you know how many amino acids we have in the body? Yeah, I'm listening. What? Yeah, that's something you get to find out. Yeah, there are several. So if you consider that there are four DNA sequences, adenine, guanine, thymine, and cytosine. And then they come in triplets. Anyone who got an A1 in maths will know that the combination of this will give you 64, like 4 raised to the power 3. That's the that's the highest number of combinatorial mathematics. If I'm to combine four things, three, like ACTG, ACT, AAC, you get 64 different recombinations. Now, but amino acids are rarely up to 64. So as a result, I'll be introducing concepts like degeneracy. Not every amino acid, not every codon, sorry, not amino acids are not up to 64. There are 64 combination of this amino, of this DNA, but not all of them codes for a polypeptide for an amino acid in the first place. That's one. But then, so if we have like take out four of them, one for methionine and then four for like the stop codons, it means that there is like 60 amino codons chasing like 22 or 23 amino acids. I forgot. I think I said the answer. I think it's 21 or 22. Does anyone know the exact number of amino acids we have? Okay. Yeah. What? Did anybody answer? I'm hoping you are writing what I'm 21?
Yes, sir.
What?
Repeat the question. Repeat the question.
Maybe someone should type it. Type the consensus so I will see. Okay, while you do that, let me go on. Yeah. So this is essentially some of the vocabularies I'll be using interchangeably or more 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 commonly in this lecture. This is just to explain what I'm going to say. Okay, I said the question, can you hear me now? The question says, how many amino acids do we have? So there's arginine, proline. What?
20. 20.
Okay, so if you remember what I said about one being the one being dedicated to methionine, which is already an amino acids, and three being dedicated to stop codons. So we have 60 different combinations, chasing how many amino acids left? 19 or 20. I think it's 21. I'm not sure it's 20. Okay, yeah, I think it's 20 amino acids. So you have 19 chasing 60. So in order to solve this, the body has what's called degeneracy built in the system. So even though there is a code for each amino acid, three or four codes can code for the same amino acid. So for example, for proline, you can have three different types of code coding for proline. In doing this, might not make sense to you, but to a mathematician, I do a lot of maths and combinatorial mathematics as part of my work. It's important to recognize that because if I'm trying to if I'm looking at a DNA sequence and I want to know what or I'm looking at a protein and I want to walk back from a protein to a DNA, it's going to be very hard. But we all do that. We still do that. There's ways to do that. It's not easy to go from amino acid to from DNA sequence to protein. But it's even much harder to go from protein to amino acid to the DNA. It's easier because if you have options of what it could be, you can actually check the DNA, the genome and find where this gene is. So if you know, so I'm trying to explain this so you understand that it's the way the whole thing is. ORF, open reading frame, codon, the set of three mRNA words that specify one amino acid. Promoters is that region which is recognized by the RNA polymerase in order to trigger transcription. Alleles, if you're thinking about diploid and haploids, one set of your genes is an allele, and that's the basis of the variation. Cistron, we're talking about polycistronic or monocistronic. So if all of the genes needed for a single process is clustered in one place, that's polycistronic. Or if they are located in different places, that's usually monocistronic. And for the record, eukaryotes Eukaryotes are monocystronic, while prokaryotes are polycystronic. When I was talking about polycystronic, I said, for example, a bacteria is basically simple in that it cannot afford to throw away a lot of its genes on multiple regions of the chromosome. So it aligns them in one particular place. So that when you are, for example, if you are transcribing the enzyme for one, it's transcribing the enzyme for every single thing. But then I will introduce that concept in the lac operon and the TRP operon. So the operons are where I'll explain this more, and that is in the regulation part of the lecture. And then obviously genes is what I've been shouting out about since morning, since we started here. So hopefully it makes sense to you guys. Does it? Any question or confusion? I'd like to know so I can explain more. Okay. So the central dogma of molecular biology, this is basically what you answered in the last question I asked, which is quite interesting. So DNA sequence undergoes transcription to give you an RNA sequence. Can you see my mouse? Let me see if I can. Okay, thank you. Okay. Uh, Sorry, I'm looking for somehow to point mouse. Okay, yeah, there's no mouse option here, so I'll just describe myself. So DNA to RNA, the concept is called transcription. So basically, you're transcribing the DNA to RNA. RNA is useful, but not that useful. RNA is basically RNA. So DNA is very stable. RNA is unstable. I'm giving you what we think about in the lab. So someone, a PhD student who is working on genes will know that he can extract DNA easily. A DNA, I can keep it in room temperature. But the moment I extract RNA, I have to put it in -80. Otherwise, it degrades very fast. So it's labile, it doesn't last. So RNA in its nature is meant to be transient. So whenever you go from DNA to RNA is basically a middleman because ultimately RNA is what is used to translate into a protein. And this entire thing I just described is basically the central dogma of molecular biology. Now, let me link it to clinical studies because this is quite important. I'm not just describing this to you. So, a change in DNA, for example, would obviously affect what happens in mRNA processing. So, for example, if you make a change in DNA and suddenly what is supposed to be a proline has joined to be an isoleucine, because, for example, you made an ACTG into an AAG, you remove, sorry, ACG into an AAG. You removed ACG to AAG. It means you've changed what it's supposed to be into what it's not supposed to be. No problem. Sometimes if you make that change, the degeneracy of the genetic code might save you. So you make a change that doesn't affect the genetic code. That's called the silent mutation. But if you make a change that changes the amino acid, so as you're lucky because amino acids have different types of amino acids. Yeah, like hydrophilic, hydrophobic, there's a charged, non-charged. So if you're lucky, what you change might belong to the same class of amino acid. That's okay. But then the big problem is where you change a charged amino acid to a hydrophobic amino acid. You change the entire behavior of the enzyme or the proteins being produced. And then that's where catastrophe starts, or you have clinical what's called response. Like diseases happen. A lot of diseases happen because there's a mutation that occurs that has been transcribed that changes that occur maybe in the gonal cells that is inherited by people, but then that's because of that change or that mutation you're making abnormal proteins or you're not making enough of proteins. So for example, can anybody think what happens if there's a change in my promoter, for example, in a very important gene? A smart person will think if there's change in my promoter, it means, for example, because my tRNA will recognize a promoter. If it comes and that promoter looks different, it might not lap on it. Or if it laps on it, it doesn't lap well. If you knock out the promoter, it means that protein is not going to be transcribed very well, or it doesn't transcribe at all. And if that protein is the protein that is responsible for for producing amylase in your mouth, a lot of people have something like that. You will die or you will not survive, or you need constant injection of amylase to make sure that what you eat is being treated. So some things that we take for granted in the human body is can be knocked out by just something. And then eventually, I think in my third lecture, I'll talk about how understanding this sort of raises social problems where some people say no to GM foods or yes to GM foods, like, oh, why are you playing God? Because we understand that if you change something, it can cause something. But then understanding biology or genetics in the way we do means actually there's no way to go from what I eat to affect my genes in the first place. So people who don't understand biology enough but think they understand it tend to overreact in terms of what happens. But if you understand it in that way, you're not only able to understand the consequence of what changes, or for example, you eating, for example, a GM cassava or a GM corn, for example. But you also understand that it has nothing to do with how your genetic makeup is structured. Those two are separate. So in this, I talked about nonsense mutation. So like I said, if if the ACTG, like the codons, you make a change in one of the codons, and rather than coding for like proline, which is an amino acid, it goes into what is called like one of the three stop codons like TAA, TCA, or TGA. So rather than you read the entire amino acid, it reads and then come there and it stops. You have what we call truncated amino acids. So we may have an amino acid, for example, the um, the amino acids used in making what's it called insulin, right? It's one of the simplest proteins we have because it was sequenced by Frederick Sanger like 80 years ago, so it's quite simple to make. A lot of diseases start by having changes that truncate that protein. So if you have a change in the codon or in any of these codon that affects or introduces a stop codon randomly into the genetic code, then you that particular change is called leads to what we call nonsense. So the protein is said to be what? Truncated. No, I didn't notice I didn't look at it. Truncated is a word where something didn't complete its natural process, right? Suddenly, maybe if you leave your medical school in your second year, you can be said to be truncated because you didn't complete the process. So if the process is supposed to make 20 amino acids and it stops at 18 because it hits a stop codon where it's not supposed to be, then that's a truncated protein. And then that could lead to physiological response that's not desirable. Now, splice site I will explain later because I think you would have to explain that from the concept of molecular central dogma of molecular biology, from going from DNA to RNA in humans. Okay, I'll explain that later. And then expression is also similar to what I explained previously, where I said a promoter there's a reason why it is called a promoter. Can anyone guess? Maybe you don't answer because I'm not sure I can hear you. It's called a promoter. So a sequence at the five prime end of I mean of an of a DNA is called a promoter. Not all the sequence. So there is like the five prime untranslated region. Then you have the promoter, and then you have some untranslated region before you have the first exon, and then you have several introns, exon, introns, and then you have five prime untranslated region. So if you're studying the genes in that way, you look at it that way. So I will encourage you to pick up a genetics textbook and read more because this is just an introduction. So I'm going to be using words that you will encounter when you're reading it too. But then, a protein is said to be expressed if if DNA sequence is successfully transcribed to an RNA and the RNA is successfully translated to protein. Anything that stops this process affects expression. So you say, is my protein expressing? So for example, I work with genetic engineers in the lab, and then they're trying to knock out a gene or to express a gene. What we usually do is we add what we call GFP, green fluorescence protein. We add the sequence of the green fluorescence protein around the sequence that I want to express. Because not every gene it has a physiological response. So I may not be able to see or maybe I'm working on a cell. So if you're working with a cell There's something called cell biology. You're supposed to read that book. But if you're working with a cell, like a cell, just a cell, and you're making changes in the cell, this or you're likely to see any change in that cell. But if you tag it with a GFP, like green fluorescence protein, with that your gene, if the cell starts to become green, you can say, oh, actually, my GFP is being expressed. So therefore, the protein I attached with it will be expressed. So understanding some of this helps you understand how we make real. So I just introduced the concept of genetic tracking, checking whether what I did works, introducing genetic engineering to it, genetic cloning. And someone who is smart will be asking, how do you even do that? It's very simple, but if you understand the process, it becomes easier to understand. Uh, find out on the thing. So I want to explain transcription mechanism. So transcription is basically the conversion of our DNA into RNA. It is simple. Let me check the time. So your RNA polymerase is this. Looks like a protein. It's a protein actually, but this is just basically a mock-up of RNA polymerase. You have a DNA. When I was talking about diploids, I said that every gene have two copies, right? You have two copies of a gene. It's called diploids, right? So this is supposed to be one chromosome. This is another chromosome. Sorry, no, sorry, yeah. So every gene has two, no, no, not chromosome, two code, two was my bad, two alleles. So this is one allele, this is another allele, and it's merged together. Now, for the transmission to happen, if the chromosome is tied together by this hydrogen bonds. So if you did in chemistry or in your biochemistry, they would have explained to you the concept of hydrogen bonds and van der Waals bonds. So, but what connects the A to T or C to G is hydrogen bonds. Now, in your chemistry, the hydrogen bonds are easy to disperse, right? You just have to apply a bit of temperature. They're not as stuck together as covalent bonds, right? You remember I just added chemistry to this. So in anything you're doing, there's always you have to apply your knowledge of something. So because these are hydrogen bonds, a cell called helicase can actually unwind this thing easily and open this thing from double-stranded to single-stranded. And then by opening up the double-stranded to single-stranded, it leaves the promoter region open for the DNA polymerase to be able to to identify those regions. And then the DNA polymerase now binds in and then drives the production the transcription of RNA. So mRNA is this RNA, or what we call messenger RNA, is basically making a copy. Transcription is basically making a copy of the original DNA. So the DNA, this is an example of DNA transcription in eukaryotes. So you have eukaryotes you have your genome within your nucleus. That DNA does not leave the nucleus. What actually leaves the nucleus is an RNA. So RNA transcription, basically in English language, transcription means basically making a copy, just like photocopying. No concept. Yeah, you put a book and then you photocopy it, right? So what this does is simply a photocopy of the gene itself. So it takes three main steps. First is initiation. So the promoter region is recognized by the RNA polymerase and it forces the DNA to open. The DNA helicase will need to open this. Next thing is the RNA begins to grow. The RNA polymerase recruits all of the NTPs, nucleotide phosphates. So these are basically lying around within the nucleus, and it recruits all of these, adds it, but it doesn't add them randomly. On an A, it adds it. On an A, it has a U. On a C, it has a G. On a G, it has a C. And on a T, it has an A. Did anybody notice what I did there? So I've talked about A, C, T, G, adenine, cytosine, guanine, threonine, thymine, right? But RNAs differ from DNA by rather than have a guanine, it has what? Maybe find it out. So a uracil replaces the, I think it's the guanine. So in this place, so rather than, because the body doesn't make guanine on its own, but it makes uracil, which looks exactly like it. In transcribing, in making that copy, it doesn't make an exact copy. It makes a copy that is recognizable by the genetic code. Is there an interesting thing going on? Okay, so the last one is the termination. Here is where once the RNA that is being transcribed is completed, the mechanism to release that RNA into the within the nucleus occurs. So that's the third step. So if I'm going to ask you, can you describe the transcription process to me? In addition to explaining what the protein does and what enzyme does what, you need to be able to articulate it in these three steps: initiation, elongation, and what? Termination. And what happens in each of those steps? Initiation, the promoter is recognized. Elongation, the RNA polymerase recruits all the ribonucleotides, and then in termination, it releases that RNA out of the RNA polymerase into within the nucleus. Does anyone have a question? Okay, so the next thing is to explain what's the difference between a eukaryotic and prokaryotic transcription. So I just described transcription as general in general terms. But then transcription occurs differently if it happens in bacteria versus if it happens in a human cell. So in a bacteria, like I said previously, bacteria does not have nuclear membranes. Like this purple thing is known as nuclear membrane. This is not present in a bacteria. So this is a bacterial cell. This is like a human cell. Talking human, it could be anything. It could be it could be like a saccharosis, like a yeast cell. Yeast cell is also a eukaryote anyway. It can be anything, simple cell. But in a human cell, we have like nuclear membrane, which which covers the DNA. Yeah. So in eukaryotic cells, the process of transcription happens within the nuclear membrane. While in prokaryotic cells, it happens in the cytosol, just generally. So I'm going to remind you what I said previously when I said prokaryotes or bacteria are designed to be safe or to be simple. Why? Because they cannot afford, they don't have clustering of their cells. I mean, what I mean clustering, they don't have like uh boundaries to their transcription mechanism. So they cannot afford to be like monocystronic, putting different genes different places. They have to conserve that energy. Now, the key difference apart from that is that if you notice, this red dot is the RNA. In a prokaryote, the RNA is made complete. It's made straight as a mRNA. mRNA means messenger ribonucleic acids. Straight, no problem. While in the eukaryotic cell, the RNA is made as what we call pre-mRNA. Pre-mRNA arises because in humans and in eukaryotic cell, RNA is rarely made on its own. First is that RNA was added with, so there's something called poly-A tail, like A sequence of A at the end. That's the poly-A tail. There's also the cap at the beginning. But in the middle also, there is what we call introns, intragenic regions, like sequences that are nonsense. They're not really nonsense, but they're just there to enable what we call alternative splicing. Notice how I talked something that might not make sense, but I'll explain that later. So if you look at, can you see my mouse, right? Not if you can see my mouse, so because I'm trying to explain something. Oh, you cannot see it. Okay, let me see if I. Okay. Can you see my? Okay, no. Okay. Sorry, I'm trying to find the spotlight. Okay. Okay, can you see my spotlight? Can you see this? Okay. So this is the mRNA, pre-mRNA. Within here is what we call the introns. This is the exons. This is the intron. This is an exon. This is an intron. This is an exon. This is an intron. This is an exon. So the exon intron interplay is what is first transcribed first. That is called a pre-mRNA. So the pre-mRNA undergoes what we call splicing. The word is splicing. Splicing is the process of removing that intron. So the intron does not code for anything. Yes? Can you repeat yourself?
What said about extron and intron?
Yes. Okay, I should repeat it, right?
What you were pointing was not clear.
So, sorry. This is an exon. This is an intron. This is an exon. This is an intron. This is an exon. So it doesn't mean, I mean, this is a schematic anyway. So the exon is what is useful. The exon, you see, is what is this color, right? The intron is like the ox blood color because it gets removed because it is useless in a way. The process of removing that is what we call splicing. I was trying to explain that. So the body undergo the gene. So DNA is able to code for both exon and intron, right? So you code it out. You have all exon and intron, alternate intron. Notice here that it does not make any intron at all in the bacteria. There's no intron. They are all exons. So there's no need for that kind of shenanigans. But in humans or in animals or anything, they make intron, exon, intron, exon, intron, exon, intron. And then the process of splicing removes these introns that are not useful. That's what I'm trying to explain. And then I talked about something called alternative splicing. Once again, if you understand maths very well, it is very likely for someone to cut this thing here and also to cut it here. You know that the gene you get from just having these two is different from if I cut it here, 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 cut it here. I'll have the full gene. But also I can also make 1, 2, 3, gene 1, 2, 3. I can make gene 1, 3, 4. I can make gene 1, 4. I can make gene 2, 3, 4. So the body has a way of using alternative splicing, cutting these introns in a smart way in order to make genes of different lengths and different purposes. So while a human or an animal cell might not be capable of being polycystronic, one gene can actually result in two different amino acids or two different enzymes or proteins. That's what I'm trying to explain, that by alternative splicing in eukaryotes, one gene can lead to several different amino acids or proteins. So that's called the concept of alternative splicing. So splicing is what removes the introns and gives rise to what we have, the exons. But alternative splicing, which means I can, in what other order, I can cut here and here, and I'll have 1, 3, 4. I can cut here and here, and then here. I'll have 1, 3, 4. I can cut in different ways and have different lengths or different combinations of exons. That process is known as alternative splicing. Go and read it. It's quite important. We still use it till today. It's very important in drug making because when there's no difference between, oh, I know this gene works because it causes this. But then if that gene decides to not work the way you think it does, what happens? So people write codes. When I mean codes, is write like programming to be able to detect or predict what kind of genes or amino acids that what kind of amino acid or enzymes this particular gene can produce. And that is one of the fundamental differences between a eukaryotic transcription and a prokaryotic transcription. Now, another thing is that after the splicing, the eukaryotes within this nucleus adds what we call poly-A tail and a cap here. The poly-A tail helps protects the protein and the amino acid, the mRNA, and guides it out, while the caps provide some protection. So they all have their own function. So let me explain that further here. Sorry. So here we have the cap I was telling you about. Look at it. It's easier to see here. And then we have exon 1, exon 2, exon 3. We have intron. This intron is useless. No one wants it. And you get what? Splicing. So you have exon 1, exon 2, exon 3 with the cap poly-A tail. This, what you're looking at here, is basically this. Basically here. Now, alternative splicing is if, for example, I decide I want exon 1 and 3, or exon 1 and 2, and I take out 3 because I cut here, and I cut here and here, rather than here and here. It means I take out 3, I have exon 1 and 2. That is the concept of alternative splicing. So, splice site mutation can cause disease. So, for example, something can happen to the recognition of this site because the intron sequence is now different. And then suddenly my spliceosome, which causes this splicing, cannot recognize it. Then it leads to a disease. So anything can lead to a disease, to a clinical disease. Anything that goes wrong in sequence can lead to a disease. What's the question?
So the mRNA stage is just for alternative splicing.
What? Repeat yourself, sorry. No, repeat yourself.
So the mRNA stage is just for alternative splicing.
Yes, exactly. No, it's just for splicing. So the pre-mRNA undergoes splicing. So any mRNA with the introns in there has not been spliced. So the process that takes it from pre-mRNA to mRNA is called splicing. It's driven by a spliceosome. But I'm saying that in clinical settings, that's in real life, because sometimes you study biology, but you also study real life. In real life, there's something called alternative splicing. That as much as there are 3 exons here, you can end up with 2 exons. You can end up with 1 and 3. You can end up with just 3. It's the body's normal way of doing things. And that process has been studied very well and is mostly used in pharmaceutical companies and biotech to study how the effect of changes in genes, how they affect physiological responses. So that's why I explained here. So the poly-A tail is for stability and help it move it to the nucleus. The splicing removes the introns. I explained that, right? And joins exons. And the cap helps support ribosomal recognition. In addition to protecting the other mRNA, which I said, it helps that ribosome to recognize it and begin protein translation. Now I will talk about the concepts of transcription inhibition. Now, even though antibiotics was discovered by mistake, by chance, the whole concept of antibiotics works because the RNA polymerase in humans and the RNA polymerase in bacteria is different. In my notes, I will explain that later. I explain that more in the notes I'm giving to you guys, where I describe why, for example, actinomycin will work on a human being and only kill the bacterial cells, and then it will leave the host. So it is because the bacterial enzyme differs markedly from our enzyme. By leveraging on that chain, that differences in, so they differ in terms of, for example, I told you there our for example, the enzyme is different. So there's 30S and then the 50S. But then apart from that, these differences in the enzyme that is responsible for transcription is what we leverage in designing drugs. So, for example, if I'm doing this drug is rarely found out on its own. No one just wake up and make it. You just throw it into a drug and like, does this drug selectively affect a bacteria. So you put a bacterial cell and you put a human cell, and then you feed them with two of those that same drug. If two of them die, you're not going to use it on a human being. No one's going to. Basically a poison. But if it kills the human cell and kills the bacterial cell, that's still a poison. That's not good. But if it only kills the bacterial cell and leaves the human cell, that's a potential antibacterial. That's a potential, what's it called? Ampiclox, actinomycin, amylitinin. So basically, selective inhibition of transcription is leveraged in a lot of our antibiotics. So in this case, I'll just giving you examples of popular antibiotics and where they target. For example, this one targets RNA polymerase, is used for treating tuberculosis. This one is an anti-cancer because it chops up DNA, but you don't just chop up any DNA. It only chops up DNA when it recognizes that that DNA is not a human DNA. And then eukaryotic RNA polymerase 2, which is basically a mushroom toxin. It causes severe liver injury anyway, but it's able to selectively affect polymerase 2, which is not present in human cells or in eukaryotic cell. So selective toxicity is the reason why we have antimicrobial targets and why antimicrobial like antibiotics work in the first place. So I'll use that I'm not sure how long we have before we finish. Okay, we have 15 more minutes, right? So I'll just use it to just give a quick evaluation of what genes, for example, you hear about genes, but actually it's a whole lot. A lot of people are using it for that. So just give you a quick story about some of my PhD where, so there's a big problem with insects chopping up all the cassava across the whole entire of Africa. And as part of my PhD with the Bill and Melinda Gates Foundation, I went across the entire Africa to collect samples of whiteflies, which were chopping up cassava. But the collection was not the main thing. The main thing was to understand what is the genetic driver enabling a particular subset of individuals to behave differently and become deleterious. Because by understanding that genetic drivers, we can now be able to design insecticides against those particular things. Because when you're trying to solve a problem, you remember the concept of the Igbo say throw away the child with the bathwater, right? As in, you don't want to throw away the child with the bathwater. So therefore, we don't want to create a drug that kills off every single insect. You want to use an insecticide that is a bit selective. But understanding that selectivity, being able to get that selectivity done, starts by understanding what differs between those two insects. So I'll introduce the concept of cryptic species. So this is quite interesting. You might not need it anyway, but you might come across it across your medical study. So those species are said to be cryptic if by looking at them, they don't look different to you. So basically, you can be looking at a baboon and a monkey, and you can't tell the difference because they look alike. But within the genes, they are different. That is exactly what I was studying. So the insects I was studying looks alike. If I look at it on the microscope, they look the same. But the only way I could tell that they were different was by sequencing what we call the mitochondrial cytochrome oxidase 1 gene. Now I'll introduce the concept of of mitochondrial genome. You know there are two types of genome in human body, right? Or so there's the nuclear genome and the mitochondrial genome. Can anyone tell me the difference? Does anyone know? Have you heard of the concept of the mitochondrial genome compared to the nuclear genome? Yeah, okay, maybe scratch that. Let me just explain that. So mitochondria is basically the powerhouse of the cell, right? Everyone knows that, right? And the mitochondria lives within this human cell. So it's usually found in the cytosol of the cell. The unique thing about mitochondria is that as much as it doesn't have a lot of genes coded in mitochondria, because mitochondria is many, within the cell, mitochondrial DNA is actually higher or a more higher proportion than the entire human gene. I mean, nuclear DNA. The naked DNA in humans is like all the 23 chromosomes, like, and the what's it called, the X and Y chromosomes, right? They are all nuclear DNA. But humans have a mitochondrial DNA separate from the human DNA. But over the years, in the past 60 or 70 years, the mitochondrial DNA has been studied mostly to help cure mitochondrial diseases. But apart from using that to cure mitochondrial diseases, they've also been mostly used for differentiating between species. One characteristic of mitochondrial genome is that it is maternally inherited. Does anyone, is that surprising? Mitochondrial genome or mitochondria, you only inherit any mitochondria where you have in your body came directly from your mother. There is no interplay, there is no variation, there's no joining, there's no mixture, there's no two, one from your dad, one from your mom. Everything you have on your mitochondria came directly from your mom. And because that can be traced, you can trace every mitochondria to the maternal genome. It's easy to use mitochondria to draw what we call genetic trees. And that is simply what I did as part of my PhD. But the understanding of DNA, not just as a chemistry, but also sequence, allowed me to be able to write programs to be able to assemble my mitochondrial sequence, use that to perform uh what's it called? To perform gene comparison, to build gene trees and use that to identify what we're dealing with and ultimately lead to better outcome for my PhD. So in conclusion, just to give you three questions from uh let's go from what we've talked so far. Enzymes that What enzymes make RNA from a DNA template? You need to know that. So there's a particular enzyme. I know I talked about the enzyme that recognize the DNA and transcribe it. Understand that. You need to be able to mention two features of eukaryotic mRNA processing. What is that difference between what's called transcription happening in the eukaryotes versus that one happening in a prokaryote? And then the third question, why can rifampicin treat bacteria without directly blocking human RNA polymerase? These are questions that I hope you use to enrich your further study, further reading, and then use that to like, okay, I think it should be thought provoking rather than just answer the question, like which enzyme make RNA. I will not just write it. I'm like, I'll describe the RNA polymerase and then what other enzymes RNA polymerase recruits. For example, I talked about DNA helicase, right? What drives the spread of the DNA into making RNA transcription? For example, I'll talk about spliceosome, talked about it, but I also talk about at what state is the DNA at every time? So DNA is rarely ever ready for transcription. What is the state it used to be? Now DNA is usually wound across a particular protein called what? Remind me. So I'm saying before a transcription happens, DNA is usually in a resting state. You need to study more to know what is that resting state. How does it look like in real life? What are the proteins that makes DNA tightly wound? And then what triggers transcription to occur? Because something needs to trigger transcription to occur. We're going to discuss that mostly, but I think you molecular genetics is quite important for that. But also in if you're doing your biochemistry studies, you'll be talking about what are the drivers of DNA transcription. Something has to occur. I'll talk about it a bit in the DNA regulation section of the lecture in terms of signal transduction, responding to environmental stress, responding to food. So if you take in particular food, the DNA that has the enzyme that digest that food will be triggered in the first place. The point is, how does the body know to make so much DNA when it needs to? There's something called feedback loop, signal transduction, but that's a biochemistry problem. So I'm trying to help you understand not just the concept, but also be able to apply that to your real life. And then hopefully you'll be able to define transcription, name the stages. I believe I said there were three stages of transcription, right? Initiation, elongation, what? Yeah, termination. Is anyone saying anything? Okay, I hope you understand. Compare prokaryote and eukaryotes and give one medical example. If you're able to do this, articulate this properly, I will have assume that you understand this lecture very well. So in the next time we meet, we'll be talking about RNA to protein. We'll be delving into genetic codes, translation of RNA into protein, and and the way inhibition of this process can come. So how do you inhibit a protein synthesis, and what drugs can be used to inhibit that, and why and how do they work when they don't work in humans? So this will be similar to what we did in lesson one, but I'll be talking more about RNA. I'll dive into why RNA is labile, why it is not stable, and why RNA is like the middleman. It's like what's it called? It's like a transient state because at any time an RNA does not last in the body. You can see proteins for a long time, you can see DNA for a long time. But if you're extracting a cell for RNA, it's a very specific process. I've done that a couple of times. It's one of the hardest things to do, getting RNA. Sometimes you have to trigger it to have a particular effect, to get that RNA you're getting. Because not every gene is transcribed all the time. So I've been introducing the concept of gene regulation in the third lecture and how genes don't just have a way of just make the body has a way of like managing its scarce resources. Making genes or making proteins is a very expensive process for the body, but the body needs to make it. But it can't make all the proteins. So what happens is that it is waiting for cues or clues, like signals, in order to make those genes. So, for example, you don't need to eat all the time because if you eat, that's an expensive process. Your body needs to tell you that you're hungry before you start eating. You can't just eat all the time because you can eat. Or so, how does the body trigger the production of enzymes and proteins that are essential for driving human life? That will be the third lecture. So hopefully I'll send you the presentation of this lecture one on Monday along with the notes on Monday. Just have a good weekend. Think about it, or you can read the molecular biology of the cell. Just go there and revamp what you understand about genes, read more. And if you have questions, I will appreciate if you type them on the next thing. Just type them. I'll go through them at the end. And I would like to answer questions because my job gives me an in-depth understanding of some of the things, not all of them, but there are some things you get from me that you probably not get from textbooks because I'm more current. I'm using some of those things we are currently using it. So textbook might be written 10 years ago, 15 years ago, but right now I'm talking about CRISPR interference. It might not be in your textbooks. You might not have heard about it. So there are things that we're using currently that's not recorded in textbooks. So those kind of things, if you find something that you think is interesting, that is relevant to what we're talking about, just collect them. Try and find out on your own first. But if you have any stump in the way, let me know. I'll help to clear that. Yeah. So I'll be sending the notes on Monday morning after I've compiled them. But this is time to ask questions if you have any question. Or you can send the question to your class rep and she can type it. I can see her message from here. Or you can log into the thing and type it. Just make sure you are mute. At the moment, we don't need anybody to say much anyway. So, okay, thanks for the question. Is there any question? Okay, please simplify as much as you can. Okay, I'll do as much as I can. Yeah. So it's the whole point is to make it easier to understand. Yeah. But I'll be using a lot of context and concepts. But anything I introduce, if you can, if for example I say something because I've been using it at work all the time and I take it for granted, feel free to tell me or raise your hands or text something, please. Uncle, can you explain what monocystronic means? I'll be happy to because I just used that and when I said alternative splicing, I was tempted to continue, but I felt like okay, yeah, maybe a lot of people might not have understood it, but that's something I work to at work. I'm like, yeah, I've looked at this sequence. I think it's like 3' alternative splicing going on. It's something I use generally at work, but Well, if I drop some words that doesn't make sense to you, let me know. Which is why I start with explaining vocabularies before I go into the context. But I'll simplify it as much as possible. So thank you. Any other question? Okay. If there are no questions, it's 5 o'clock on the dot. So we'll meet next time. I'll communicate that to the class rep when the next lecture will be. But I'll send the lecture notes and the PowerPoint presentation to the lecture to the class rep on Monday. And then the lecture notes will be various. So it will be enough for you to pass, but also use that as a stepping stone to do more on your own. Yeah. Thank you very much. Yeah. Have a nice day, everyone. Yeah. Bye.